We're going to move on to the next section now, just before lunch. So we're going to be hearing from our ASH abstract winners. First of all, I'm going to invite up Dr. Yakinthi Chrysokoidou. Yakinthi was awarded BSc in Biological Applications Technology at the University of Ioannina in Greece in 2015, subsequently an MRes in Biomedical Sciences and Translational Medicine at the University of Liverpool, went to Imperial College for a PhD looking at mechanisms producing fibrosis around mesothelioma, and she then joined the Institute for Cancer Research, working on intrinsic and extrinsic mechanisms of IMiD resistance in multiple myeloma, and she's going to introduce her talk now.
Hi everyone, thank you for the introduction. I will be speaking to you today about our project investigating the impact of cereblon mutations on response to IMiD and CELMoD agents in myeloma. So we all know our immunomodulatory drugs, or IMiDs. They are a standard of care at all lines of myeloma therapy, used in combination therapies at multiple stages of the disease, and also as a single agent maintenance therapy. We have our IMiDs that are currently used in the clinic, lenalidomide and pomalidomide, and then we have the next generation IMiDs, also referred to as CELMoDs, iberdomide and mezigdomide, which are currently in clinical trials. These compounds all function in myeloma through the same mechanism of action. IMiDs target cereblon, the substrate receptor of the CAL4E3 ubiquitin ligase, and when cereblon is bound by IMiDs, its substrate specificity is altered, leading to ubiquitination and degradation of new substrates, including transcription factors Icarus and Aiolos, which in turn result in downregulation of IRF4, a key transcription factor in the survival of plasma cells. Through sequencing of patient biopsy samples, we and others have discovered that post-IMiD treatment, approximately 1/3 of patients presents with genetic alterations in cereblon. And while it might be easier to predict the effects of whole exon splicing or copy loss in cereblon activity, the functional effects of the missense mutation are less predictable. And as sequencing panels become more widely available as part of myeloma patient diagnostics, it's of increasing importance to understand the functional impact of any mutations identified, so we sought out to investigate these missense mutations functionally. So, from every publication that recorded cereblon mutations in patient datasets post-IMiD treatment, we identified 34 mutations, those highlighted up at the top. And from those, any frame shift or stop codon mutations that naturally result in non-functional cereblon have been excluded. Then we selected 11 missense mutations for further investigation, deliberately choosing mutations spread through the entirety of cereblon. On the left, you can see how they line up through the entirety of cereblon. There are some in the lone binding domain, this blue segment where cereblon interacts with another protein in the E3 ligase complex, DDB1. And in the purple here, we see the mutations located in the thalidomide binding domain, where the drug actually binds. And within that, we have zinc ion binding sites highlighted in teal. And in red, we have the beta hairpin sensor loop that mediates cereblon switch between open and closed conformation. On the right, you can see here each mutation mapped in cereblon. These are the mutations mapped in pink, and the teal molecule up there is lenalidomide. So we sought out to investigate the impact of each one of these mutations. The first step was to generate a cereblon knockout model, meaning a cell line that does not express the protein. So we started with MM1S, an inherently IMiD-sensitive cell line. Those cells responded to treatment. And then we CRISPR'd out cereblon. 2 cell lines were generated, and you can see they're highlighted here in red and blue. And as you can see, these cell lines do not respond to treatment, both to IMiDs and CELMoDs. And of course, we lose all the downstream effect. We don't see degradation of the neosubstrates or downregulation of IRF4 and cell death. And then next, these cereblon knockout cells were transduced with a plasmid expressing cereblon, but each one had a point mutation, mimicking the amino acid chains that is observed in patients. And the first observation is that reintroducing cereblon back into the cells does result in cell death, and the cells are resensitized to IMiDs. So after that, we aim to understand which missense mutations are deleterious to cerebellum function and whether any mutations are associated with the differential response between image and cell modes. This was done through 2 types of experiments aimed at ascertaining the cell's viability response to our image and cell modes, and then functional validation for each mutated cerebellum to observe its activity. This heat map consolidates the results of 240 cell viability assays across all cell lines with cerebellum re-expression for each mutation against image LEN and POM and cell modes IBER and MESI, and the viability responses match the neo-substrate degradation results shown by Western blot, and I'll be showing you examples of those going forward. But we can clearly see that the viability responses are divided into 3 major categories. First, we have the mutations that do not alter cerebellum function. These behave like re-expressing wild-type cerebellum, and you can see here in the heat map where cerebellum is highlighted, there is a proper reduction in cell viability as expected, both to image and cell modes, and matching that, we have degradation of neo-substrates and downregulation of IR4. I've highlighted all the mutations that behave just like in this manner, just like wild-type cerebellum, and we can see that most of them are located in the lone binding domain, and surprisingly, even mutations in the thalidomide binding domain, here and here, they can be tolerated as long as they do not disturb the folding or stability of cerebellum. These do not appear to alter the structure in any way enough to result to image and cell mode resistance, so they are tolerated to result in a functional cerebellum and response to treatment. The second category are mutations that completely abolish cerebellum function across all drugs, both image and cell modes. As you can see here in the heat map, or negative control containing only an empty vector. This response is all red values. Accordingly, there is no reduction in cell viability, and there is no degradation response or cell death. And in this category, we have our positive control mutation here, mutating this tryptophan. That is a part of a tritryptophan pocket. That's the exact spot where imides bind within the thalidomide binding domain. And mutation of any one of these molecules leads to structure collapse and non-functional cerebellum. In this category, we also have one additional mutation at position 397, and mutation of this histidine at position 397 into a bulkier tyrosine cannot be spatially accommodated and is also predicted to lead to complete collapse of the tritryptophan pocket, as seen here, which would prevent any imide or cell mode binding. And indeed, again, you can see there is no degradation response or reduction in cell viability. The third and final category are mutations that have an intermediate effect. These mutations, shown here in these 3D models, occur in amino acids that are predicted to be critical for the proper folding and stabilization required for active cerebellum. The common element between these 4 mutations is that there is a differential response between image and cell modes, and mezigdomide in particular was able to elicit a cell viability response in every case. For example, here, this mutation at amino acid 326 appeared to induce resistance to image with much reduced cell viability response to them, with much reduced cell viability response and neo-substrate degradation. However, we can see here that cell modes still induce neo-substrate degradation and reduction in cell viability, albeit to a lesser extent than seen with wild-type cerebellum. The mutated molecule here is near a zinc binding site, and any alterations here are expected to impede the proper folding and stabilization required for active cerebellum. However, this appears to have been overcome with cell modes. In order to further validate our results, we performed dynamic modeling where we stimulated the interaction between cerebellum, lenalidomide, and Icaros, the neo-substrate, for 100 nanoseconds to observe their interactions in time. This is the result of that work, which clearly shows that the change of this proline to this serine results in physical clash of the structures that does not allow for the proper folding formation required for cerebellum activity. And although we can't model yet the binding of mezigdomide in such a manner, Due to the lack of a reliable crystal structure, it is our hypothesis that mezigdomide, by bridging the amino and carboxy terminals in cereblon, it's forcing it into its closed active conformation, and even if that occurs to a small proportion of cereblon complexes, it is still sufficient to induce a degradation response and myeloma cell death. This may make the use of CELMoDs in patients whose myeloma harbors these mutations viable even after the development of IMiD resistance. In summary, we identified 3 different types of patterns of response to IMiDs and CELMoDs with mutations at different sites in cereblon. Here, the mutations that appear to leave cereblon active for both IMiDs and CELMoDs, mutations that interfere with structural elements leading to IMiD resistance, yet activity is retained with CELMoDs, and then mutations that lead to no response to IMiDs or CELMoDs, and those are the ones that are predicted to lead to collapse of the tritryptophan pocket. These results may have important implications for the interpretation of cereblon sequencing results from patients for future therapy decisions, particularly differentiating for those who may, despite relapsing on IMiDs with cereblon mutations, have the potential to still benefit from the use of CELMoD agents. And I'd like to finish by thanking the other members of our team, our founders, and the UK Myeloma Society and GSK for supporting our research. Thank you for your attention.
Questions from within the room? Gui.
Thank you. Very nice talk. Very nice work. Do you? But if one were to try and use to try and design a biomarker to select patients for treatment with, say, lenalidomide or mezigdomide, I know you can stain bone marrow biopsies for cereblon, and you can measure protein levels of cereblon. Do you have any idea, any information on whether this mutation changes the epitopes that antibodies bind to?
Unfortunately not, because these are single nucleotide missense mutations. Usually the antibodies will still probably bind anyway. So this would only be usable after sequencing results, ideally.
So staining wouldn't help, no?
Probably not.
Okay, well, thank you very much. Oh, sorry, Niels.
Very nice work indeed.
Thank you.
Maybe 2 questions, if I may.
Of course.
Have you already looked at samples post-LEN, post-POM, post-IBR, post-MESI, and do you see an accumulation of what the mutations that you predict to be seeing?
So these are not mutations that we predict. These are mutations that have been seen in patients post-treatment. Unfortunately, most of them have been seen once or twice because we don't have a lot of sequencing data from patients, but these have appeared post-treatment, usually with LEN and POM, as we don't have any data for MESI.
Okay, you didn't look at the samples post-IBR, post-MESI to see whether you see an increased rate of such?
No, there isn't enough sequencing of patients yet.
And the second question is, well, IMiD CELMoDs have also immune-mediated effects. So do you think that cereblon mutations might also occur in healthy immune cells because of all the pressure that you give, or this has never been seen in your data set?
This is what we are currently working on. So I guess, moving on from myeloma cells as they are and taking patient samples and isolating immune cells to actually see the effects, but nothing has been established yet.
Okay, thank you.
Thank you.
That's great. Thank you very much.
patients who do poorly. Often these patients are only realised to have been high risk in retrospect and may have warranted more intense frontline therapy. There is a need to identify these patients at diagnosis so they can be treated separately to improve outcomes. Now, a variety of approaches have been used to identify high-risk patients, including FISH and the Sky92 gene expression panel. There is therefore the opportunity to continue to refine this to improve diagnostics and to better treat myeloma at diagnosis. At the genomic level, there exists a variety of methods that could be used to identify high-risk markers. Whole genome or exome sequencing would capture nearly all variants, but this remains expensive and requires significant time and resources for interpretation and are hard to scale. There is the opportunity to capture the most important myeloma-specific regions in a more focused method through targeted sequencing. This has the advantage of being more cost-effective and scalable with potentially greater sequencing depth. Recently, the Myeloma Genome Project, or MGP, targeted sequencing panel was created as a collaboration between Oxford and Indiana University and scientists at Bristol Myers Squibb. The schematic highlights that the MGP panel captures almost 300 loci that are recurrently mutated in myeloma. Precise FISH probes vary between centres, but for comparison the yellow stars highlight only a handful of regions that are currently captured by FISH under UK reimbursement. During development, the technical performance of the MGP panel was benchmarked against whole genome sequencing. Importantly, some of these markers, such as MYC rearrangements, may have prognostic value and are currently missed by FISH. In the current study, we aim to explore the potential clinical utility of the MGP panel. We did a head-to-head comparison with FISH in newly diagnosed myeloma patients. Our goals were to conduct a pilot study to implement its use within the existing framework of a National Health Service lab in the UK. We were interested in how the extra resolution afforded by the panel would alter risk stratification and treatment decisions at diagnosis. Our pilot study included 46 newly diagnosed myeloma patients. We did knowingly pre-select some patients who were classified as standard risk at diagnosis, but we know did poorly. Each patient had both FISH and the MGP panel attempted from their diagnostic bone marrow aspirates. We found that the MGP panel had a greater rate of successful reporting. Around 20% of patients do not have FISH reported due to technical failure. By comparison, the panel failed to be reported for just 1 of 46 patients. To assess how this would alter treatment decisions, we conducted a simulated MDT meeting. This was composed of 10 consultant haematologists who specialised in myeloma, and all were blinded from each other. The MDT were presented with each of the 46 patients' cases twice, once with the FISH report and once with the MGP panel, but cases were randomised to avoid recall bias. As expected, we found that the MGP panel captures many more genomic markers than FISH. The MGP panel reported nearly all variants detected by FISH, and additionally reported equivalent loci at greater resolution. For example, the MGP panel caught subclonal copy number changes, including 1q gain in an additional 5% of patients, and 17p loss in 17%, that were missed by FISH. In other cases, the MGP panel highlighted prognostic loci not captured by FISH altogether, such as MYC rearrangements in 35% of patients. In the simulated MDT, basic clinical details were provided for patients, based on what was known at the time of diagnosis. The right panel displayed either FISH or MGP panel report. In cases where FISH or the MGP panel failed, the right panel simply stated that the test failed, as would be observed in clinical practice. This is an example of a case with FISH. The clinicians would also have seen the same patient again with the MGP panel report instead, as shown. The MGP panel report summarised the single nucleotide variants, the copy number changes, and translocations captured by the panel. This is a case of a patient who had a normal FISH report, but where the MGP panel was able to identify a MYC translocation, for example. Each MDT panelist were asked the same set of questions for each case. Clinicians had to designate the patient as standard or high risk, and were asked whether they would opt for an intensified regimen if available. If there were therapeutically actionable variants, as per the My Drug trial, panelists were additionally asked whether they would consider prescribing these in addition to standard induction therapy, if available. For transplant-eligible patients, clinicians were asked to choose between single or tandem transplant, and as for all patients, whether they would continue maintenance therapy for a fixed duration or continuously to relapse. For each case, we took the consensus majority MDT view shared by more than 50% of panelists for each of these decisions and compared consensus decisions between FISH and the MGP panel. Our pilot study found that the MGP panel altered clinician risk designation. With FISH, the MDT classified 13 patients as high risk, but with the MGP panel, the same clinicians reclassified an additional 13 patients from standard to high risk. We then looked at these upclassified patients and asked what were their associated genomic features revealed by the MGP panel. These patients were enriched for prognostically significant copy number changes and MIC events. Therefore, the additional genomic resolution available through the panel can identify prognostic markers that are associated with changes in clinician-led risk stratification at diagnosis. Next, we considered how treatment decisions change with the addition of the panel information. We found that 24% of patients were reclassified from a standard to an intensified regimen of induction chemotherapy with the panel. Of 25 patients who were transplant eligible, an extra 3 patients, or 12% overall, were reclassified to receive a tandem autologous transplant with the panel. Our pilot study therefore finds that the MGP panel may enable risk-adapted tailoring of the initial treatment pathway for newly diagnosed patients. Finally, we found that the MGP panel highlights variants that may be therapeutically actionable. There is the ongoing MyDrug trial, which is piloting whether existing targeted therapies can be repurposed for specific variants in myeloma patients. We assessed whether patients had any one of these 4 variants, which would qualify them for a targeted therapy. In eligible cases, clinicians were asked whether they would consider a targeted agent in addition to standard induction therapy. Clinicians stated that they would have considered a targeted agent in the majority of cases if the option were accessible. So we find that the MGP panel has the potential to highlight novel actionable therapeutic insights. Our next steps are threefold. Firstly, we plan to roll out the MGP panel to profile all newly diagnosed myeloma patients locally. Secondly, we plan to use the RADAR trial of FISH-directed treatment intensification to do a head-to-head comparison between FISH and the MGP panel in a much larger and unbiased cohort. Thirdly, we envisage building an observational registry where the MGP panel is integrated for all UK myeloma patients. In summary, we did a head-to-head comparison between FISH and the MGP panel, finding that the panel has a higher technical success rate and that it reports several prognostically significant variants that are currently missed by FISH. At a similar price, we believe that the panel offers potential to empower clinicians to tailor clinical decision-making for newly diagnosed patients. This work has been highly collaborative across centres, so I'd like to thank all involved and our collaborators. Thank you very much for listening, and I'll gladly take any questions.
Okay, so we're just waiting. Gaurav, are you with us online? Hope so. I have a question. Yes, here he is. Hi, Gaurav. Welcome to UKMS. And thank you very much for your excellent talk, which we've just enjoyed.
Thank you.
Thanks. So I have a question about sample quality and how much that is important in success rate of this technique. Do you think it's something that could be widely applicable in the UK? I assume because you can go back and use retrospective samples from myeloma 15, it's not that hard, but can you comment on that versus FISH, which we all know is fraught with difficulties?
Yes, we did have a higher technical success rate with the panel. These were samples which were between 3 and 4 years old that were biovamped, so from about 2019 to 2020. And they performed well, so we'd envisage that this would have high success rate in radar samples, for example.
And how much material do you need? And is it DNA only, or do you need RNA as well?
So this is DNA only, and exact quantities I don't have off the top of my head,
Okay.
but we managed to have enough for all the patients that we wanted to profile.
Great. Okay, any further questions in the room? Kui has a question. So Professor Kui Yong from UCL.
Hi, Gara. Very nice talk. I wanted to pick up on the issue of using targeted treatment. This is out of the scope of your abstract, of course, but where would you envisage in the treatment pathway one would use these agents? And would they be different depending on what the target was?
Yeah, that's a really good question. We asked the panelists whether they would include them at diagnosis, and in addition to any standard induction therapy they would have used anyway. So not means there's a replacement. In terms of where in the pathway, I don't have a clear sense, as I'm not a myeloma clinician myself, but I think it'd be really interesting to explore that in future work.
Thank you.
Great. Okay, we've got a question from the online audience. So the questioner says, it makes one feel very uneasy not to have FISH at diagnosis for young patients. Is this a viable alternative, and how long before it's routinely available? Along the lines of what I was asking.
So we'd hope it's not too long before it might be available. It's at a similar price to FISH. So there's the question of what we can do with all the extra information, and clearly one of the limitations is there's a lot that we need to still understand about what is prognostic and what is not. But at the very least, having an assay which might have higher technical success rate may be really good for these younger patients, as the person mentions. But we do need to do further work in larger sample sizes and to fully do a head-to-head comparison in a less biased cohort.
Sure. Okay, great. Any further questions? Yeah, we have one at the back.
Hi, I was just wondering whether in the future it would be possible for this to be used as a blood test as opposed to requiring a bone marrow sample?
Potentially. I think, as far as I'm aware, there is a field looking at trying to ascertain these myeloma mutations from peripheral blood with liquid biopsy. I think there are a lot of technical challenges still in the current stage of that, although it would be lucrative to be able to do that in the future.
Yeah, I guess it would depend on the sort of density of circulating tumour cells. Any further questions? No? Okay. Well, once again, thank you for your excellent talk. Thank you for joining us online for comments. And that would conclude the ASH bursaries section. We have run a little bit over time. We were planned to rejoin at 1:25. We can extend that to 1:30, I'm told, so please be back by 1:30 for our Michael Morley speaker. Thank you.